So good morning, and we were gonna move on to examination 3, lecture 1, which is introduction of metabolism. These slides, I'll just go over it. Then during the lab section, because we'll do lots of the biochemistry test. So during the lab section, I'm gonna draw on the blackboard. That also will be a supplement for the lecture. And then I will also have a separate video on the YouTube, a post on the YouTube regarding the calculation of the 38 ATP generated through the aerobic respiration and something else. So we go over these slides first. So when we talk about metabolism of the bacteria, we are actually talking about two opposite directions, which is catabolism and anabolism. So what is catabolism? Which means a large complex molecular. You break down, you degradation, become a simple molecular. Very important, catabolism release energy, generate energy. So we call it an exogonic process. Now, of course, you know the energy is ATP. Anabolism, what is anabolism? By the opposite direction, the biosynthesis of complex molecules from simple compounds. That is, we call it endogonic. Therefore, we using energy, so consuming energy. Now, where is the energy comes from? The energy comes from catabolism. So remember, metabolism is a combination of catabolism and anabolism, always coupled together during the short lifetime of bacteria. If one of them has been stopped, the bacteria will be died. That's why one of the way to prevent bacterial growth is to stop the metabolic process. Okay, so this is another picture which is showing catabolism and anabolism. You can see we have, let's say, glucose. If catabolism, we generate energy, then we have a waste products generated, like acid carbon dioxide, which is a gas. So based on these waste products, we could see the color change in the bacterial media. We can do a biochemistry test. Now, anabolism, bacteria can use energy, like nitrogen, sulfide, that's nutrients. Then they get ATP. The ATP is from catabolism. Then have a subunits like amino acid, nucleic acid, lipids. Then composed become a cell wall structure, membrane, nucleic acids, ribosome, and surface structures. So that's called anabolism. Here we want to mention a little bit about two laws of thermodynamics. Now, it's actually very easy to understand. What is the first law of thermodynamics? Which means the energy is never created, never destroyed, it is only transferred from one form to another form. The second, which is called entropy, which means the amount of the disorder in a system. So said very easy, what is the second law? The whole system of the bacterial metabolic process is always from order to disorder. So the amount of disorder is always increased. During their short time of the life, bacteria try to make it organized, but the disorganized disorder is always a tendency. At the end of the day, the disorder is too much. You cannot put it back to a normal life. The bacteria will be died. So those are, I use those to a layman language, which is explain the first law and the second law of thermodynamics. Now, what is the energy currency? Everybody knows called ATP, which is called adenosine 5-triphosphate. So we have adenine, we have a ribose 5-carbon sugar, then we have a phosphate right here. Now the first one is AMP. This one only one phosphate. The second one is ADP, which is one high energy phosphate. Another one is ATP. We have two high energy phosphate. And these bonds are actually very loose attached. They are easily to be broken to generate ATP that can be used. So the reaction is always AMP go to ADP becomes ATP. Of course, it's reversible. But always remember, I mentioned, if AMP do not become ATP and ADP and ATP, they cyclic become CAMP, then all the bad things could happen because it's like the money has been locked in your wallet and you cannot use energy to do the process. Okay, so that is ATP. Now here, what we're going to talk about is oxidation reduction reaction. Now, I want to let you know, most of the metabolic process is oxidation reduction reaction, which means it's related to the electron transfer. So electron is always transferred from donor to acceptor. And how do we know they have more energy? If something has more electron or more molecular, then more energy it will has. So electron transferring actually represents energy transfer. This is something you learned in your chemistry, but some people always confused. What is called oxidation? You lose electron. Now this is very easy. You look at a 90 years old grandpa's face. Not much moisture, not much energy. Old people's face, that's oxidation, lost electron. When you look at the 20 years of the young lady's face, well, it's a lots of energy there. Why? Because lots of electron. Now, I always tell you how to remember this terminology. If you go to graduate school, you need to taking a exam called GRE. So reduction gain electron, we say GRE, and R is print, take it out. So reduction is gain electron, oxidation lose electron. Gain electron, which means more energy. Always remember 20 years old young lady's face. Okay, just give you example, you easily to understand. Now, what means oxidation? When you lose electron, that product is oxidized. When you gain electron and that product is reduced. But you also should know compound A, because they contribute electrons, so compound A called reducing agent. And the compound B, we call it oxidizing agent. Okay, so these are the chemistry you should know. Now, how do you know one chemical is a good electron donor? Another chemical is a good electron acceptor. How do we know that? It's very easy. We do a two-half reaction. This is a two-half reaction, which means we tested their voltage. The more negative, which represent, indicate it is more potential to be a good electron donor. The more positive, which means it pretends to be a good electron acceptor. This is also the reason why the order of the electron transport chain is always coming from FAD, NADPH, then finally is go to the oxygen. Because you can see NAD, the volts value is very negative, minus 0.3. And oxygen is very positive, 0.82. So NAD is always pretended to be a good electron donor, and oxygen is an electron acceptor. That is why oxygen is always the last component of the electron transport chain if it's aerobic respiration. Okay, so that's called a two-half reaction. Now, I just mentioned there is an equivalent constant for the oxidation reduction Rhodex. More negative is a better electron donor. More positive is a better electron acceptor. That's why you will see the electron transport chain sometimes it looks like this. So at the beginning is more minus, at the later is more positive. Now how do we know there is a more energy generated through one metabolic pathway compared to the other? We want to see the difference between the beginning and the end. If the difference of the voltage is larger, which means more energy going to generate. So let's say the electron transport chain is like this. Let's say the electron is coming from glucose to oxygen. So what's the difference between that? That is about 1.5 volts, because at the beginning minus 0.5, at the later 1.0. So difference is 1.5. So let's say we have another electron transport chain, which is coming from pyruvate to oxygen. Then what's the difference? The voltage difference is 1.1 compared to 1.5. So you will see from the glucose to oxygen, it's generated more energy compared to the pyruvate to go to oxygen. Because the greater the difference between the E0 of the donor and the acceptor, the more energy going to generate. Now, where are the electron transport chain? Okay, so you need to know the electron transport chain for bacteria, it is always at the cell membrane, plasma membrane. But for eukaryotes, let's say yeast and the fungi, they're at mitochondrial membrane. So you should know this is the first difference. Now the second, I will draw on the blackboard what a typical electron transport chain. But remember, this is always coming from the first chemical, which is more negative, because there's a potential more energy stored there. And the last chemical is always more positive because it's pretend to be accept electron. Okay. Now during the electron transport chain, we need lots of them to carry electron, to transfer them. Therefore, we call it an electron carrier. Now, what are they? They are NAD, NADP, and others. So here are the example of a typical electron carrier. NAD, we call it nicotinic adenine dinucleotide. NADP is nicotinic adenine dinucleotide phosphates, and FAD is fulvine adenine dinucleotides. Now, very important information here. NAD and FAD is used during catabolism process. They will be promote a protein motive force, and I mentioned earlier, will be trigger ATP synthesis. So these two actually are generated through the and catabolism process, but NADPH is different. This is going through biosynthesis. So NADPH is actually going through an anabolism process. Now, what is a good example is Calvin cycle. They involved NADPH. Okay, so here's something we will mention later on. Now, how many ATP going to generate during NAD reduced to NADH? It is 3 ATPs. How many ATP generated from FAD to FADH2? It's 2 ATPs. This rule is old. Right now, you learn in your biochemistry class, you will say NAD reduced to NADH is 2.5 ATP. FAD reduced to FADH2 is 1.5 ATP. Why? Because we take consideration of the ratio between oxygen and the phosphate. Now, all these I'll draw on the blackboard later to explain it. Now, we have a lots of the metabolic pathway in a biochemistry cycle during the bacteria's short lifetime. Okay, so we have a linear. This is like lactose fermentation process. We could be branched. We have a starting compound, intermediate, and 2 different end products. This is kind of like glycolysis. The last one, it could be a cycle of the metabolic pathway. We have lots of intermediate product, have end product. This is kind of like citric cycle. Okay, we're gonna all mention these later on detail. Now, we want all these metabolic pathway to happen efficiently, which means they're gonna happen in a very short period of time. Therefore, we need an enzyme. And you learned about this already, so we're gonna go these real quick. So what is enzyme? First of all, enzyme is a protein. Second, enzyme is very specific for the substrate. Only one type of the substrate, they can be recognized. Enzymes, they were not involved in the reaction to the end products. They have a very high reaction efficiency. Lots of the time they require coenzyme activity, and it is heat sensitive. But I want to be cautious here. Most of the enzymes are heat sensitive, but there is one enzyme is not heat sensitive. Actually, it's heat resistant, which is Taq enzyme. The enzyme used in the PCR process, polymerase chain reaction, because that enzyme is coming from geobacillus, and geobacillus is a thermophile. So you should know about that. Not all of them are heat sensitive. Most of them are heat sensitive. Now, how do you know it is an enzyme? Every time when you see ASE, it is an enzyme. For example, catalase, urease, oxidase. You have a suffix ASE, which means it's an enzyme. Usually it's named by the substrate. Now, why enzyme could be have a more efficiency for the biochemistry reaction? I'm not going to go very detailed. You're going to learn about chemistry class. But the bottom line you should know is to decrease the energy required for the biochemical reaction. So you see this blue line, the AB becomes CD. If there is no energy, if there is no enzyme, it require higher free energy to happen, taking longer time. But when you add enzymes, the energy is decreased. So less enzyme required for bacteria to use for the biochemistry reaction. That's why it happened more efficiently. Now, enzyme is not really very easy. Usually it's a good combination. So the protein, we mentioned enzyme, most of them is a protein. The protein part, we call it ample enzyme. Now, lots of them, they need a helper. So we call it a cofactor. The cofactor, usually it's a firmly attached. We call it closetic group. If it's loosely attached, only a carrier or like a shuttle bus, we call it a coenzyme. And the ample enzyme and the cofactor combined, we call it a hollow enzyme. I'll give you an example later on. Now, enzyme is always very specific for one type of the substrate. So this model is very typical. You learned in your biochemistry class. This is called key lock model. So you have an enzyme, there's the activation site. There's the substrate, very fit for the position. So it attached and the biochemistry reaction happened. And once the biochemistry reaction finished, then the products released, and then the enzyme will not be changed, which means they're not going to be involved in the biochemistry reaction. That's called the key lock model. Now, what are the conditions will effective enzyme? That's very simple. Number one, temperature. Do you remember most of the bacteria we grow at 37 degrees Celsius? Same as human being. Your normal temperature, 36, 37 degrees Celsius. Because at that time, the enzyme activity is most proficient and the most active. When you have a fever, you're not going to eat too much. You're not going to have a good aptitude because the metabolic enzyme will be affected by the temperature. Even a 0.5 degrees Celsius will be very sensitive for the enzyme. Second, the pH. Do you remember acidophile, alkaline, neutrophile? Most of the bacteria, the optimal pH is 7.0. Now, of course, enzyme, we always talk about there is a called a feedback reaction. So it will be affected also by the substrate concentration, the products, and the enzyme concentration. Now, there is a competitive inhibition to impact the enzyme. Okay, we already mentioned there is a sulfur drug called a sulfonamide. We just call it sulfur drug. This is the first made antibiotics, but we still say it's antibiotics. So what's the story? Bacteria cannot to form folic acids. They need some of the reaction. They need a precursor called a PABA. So the PABA is with enzyme reacted, then form folic acids. Now you have a sulfur drug. The morphology of the sulfur drug to PABA is very similar. So instead of PABA attached to the enzyme, the sulfur drug attached to it. So they have a competitor. Therefore, PABA cannot attach to the enzyme, so the reaction does not happen. Then the folic acid does not generate. Some of the bacteria will be inhibited to grow. So this is called the competitive inhibition. Now, the competitive inhibition, you always remember, which means the morphology of the enzyme and the morphology of the inhibitor are very similar. So they can be competitive to grab the place where there happens the biochemistry reaction. But most of the time, you should know, it's have the other way to do. Some of the inhibitors, they're not really to be competitive. Instead of they grab the place, happen the reaction, they'll attach to the side of the enzyme. They will change the enzyme morphology. Therefore, the substrate and the enzyme cannot be combined to have the reaction. A good example is the lactose operon, and we will mention in the examination 3. And I also want to mention lots of the time, the end products will have feedback effects for the enzyme. Because in a very short period of time, bacteria needs to be make sure the biochemistry reaction is efficient. If too much products generated, they will say, please stop reaction. Because if you generate more product, you're wasting. You're wasting time and energy. So end products will be impacted the enzyme due to the metabolic pathway. These we call it allosteric inhibitor. Now this is another one, which is give you a picture to clearly show you the feedback inhibition, which is coming from the end products. The end products could be reacted to the intermediate product, or they could also be affect the beginning of the substrate. It depends on different situation. Okay, so this is the end of this here, which is gave you a very brief opening remarks, and more will come in, will be drawn on the blackboard.